Very nice to be here and very happy to see such a group of young scientists working in malaria from really around the world. What I'm going to talk to you about is how we can think about using vaccines in the effort to eradicate malaria. And what I'll do is really, I'm not going to share data, is to really think about concepts. Think about what we have been doing so far and what needs to change if we are to use vaccines for the eradication effort. So to take a look at what we have been doing so far, well, the sort of picture people had of malaria over the last 20 years when people started working over the last 20, 30 years trying to develop vaccines, what we had was, oops. So we had, this shows you the endemic regions of malaria for falciparum and vivax. Typically we had about 500 million cases of falciparum with around 1 million deaths. So that is what people really focused on. That is a major problem. These were primarily deaths in young children and primarily in Africa. So there was clearly an important need to try and protect these young children from dying of falciparum malaria. There's also vivax malaria, primarily outside Africa. And although originally vivax was always thought of as being the benign form of malaria, more recent studies have shown that vivax can cause severe malaria, although not as many deaths as from falciparum. And over the last 10 or so years, with intensified use of currently available malaria control tools, as you heard in the previous two talks, which is combination therapy, insecticide-treated nets, and indoor residual spraying, you can actually drive down malaria transmission rates and malaria incidence quite well. And as you saw in the slide from Lee, this number, which was sort of typical what we were over the last 10 years, this has been driven down to, if you remember on Lee's talk, this number is about half of about 200 million cases in 2012 and about 600,000 deaths. So we have made an impact. However, the question is, these tools require that you constantly keep using them. And so the question is this really sustainable in the long term? And that's where the role of a vaccine could be valuable. A vaccine could be something that you could use, especially a vaccine that as we will see, but we need to think of some different kinds of vaccines compared to what we've been trying to develop so far. We could actually make this process in addition to these other tools. So vaccine alone, again, is not going to do it, but it could make these other tools effective in the long run and more sustainable. So given that scenario, what we've been doing so far, as I mentioned, the focus has really been on reducing morbidity and mortality due to falciparum. And that was fair enough, given the number of children, as I saw, more than 1 million children dying every year around the world, primarily in Africa. So that was a perfectly reasonable goal to try and do something about, which is reduce morbidity and mortality due to falciparum. And so the vaccine was primarily for use in the group where this mortality occurs. So the target for this vaccine was always thought of as being young children, infants and young children. And to achieve that, the focus has really been on pre-erythrocytic and asexual stage vaccines. You will recall that all the clinical symptoms of malaria are due to the blood stage. So it's important, if you are going to have an impact on morbidity, to do something about the blood stage. And you can actually, if you have a very effective pre-erythrocytic vaccine, you can actually also not allow blood stages to develop in the first place. So the focus has been, in terms of making vaccines that will reduce morbidity and mortality, the focus has been on developing pre-erythrocytic and asexual stage vaccines. And in 2006, WHO with other funders came up with a malaria vaccine technology roadmap, and where they said as a landmark goal, we should try and attempt to make a vaccine for falciparum with 50% efficacy by 2015. And that's next year. And you will see, as I'll show in my next slide, we are close to achieving this. And as a longer term goal, they set out the goal as developing a vaccine for falciparum with 80% efficacy by 2025. And that's the job of you guys, the young people in the audience, to try and achieve. Now, given this, these were the priorities. What was lower in terms of priority was a vaccine to reduce malaria transmission. There has been basic research, very nice work coming from a number of labs showing that you can, as I will show you, make vaccines that will elicit antibodies against sexual stage or mosquito stage antigens and block transmission of malaria parasites and not allow parasites to develop in the mosquito. However, funding and the sort of focus on trying to develop these vaccines had a lower priority over this previous 10 to 20 year period. And another area that again took lower priority was addressing the problem of vivax. The focus was primarily on falciparum. And these are the two things that will need to change if we are to really adopt the eradication agenda. Now, why do we think that a malaria vaccine can be made? Why do we think a malaria vaccine is feasible? Well, here are the reasons. First of all, if you, and this is sort of data from a number of sites around the world, what you see in terms of this, what this plots is incidence versus age in years in a malaria endemic region. The green curve shows you the incidence of severe malaria. So severe malaria primarily peaks in very young children, in sort of the 2 to 5 year age group. What that means is that as the children grow up, as the children grow, they do develop, first of all, immunity to severe malaria. Next, the gray curve shows you the incidence of clinical malaria, mild malaria. And there again, you see a peak. The peak is a little shifted to the right. But again, it's basically children around 5 years of age have, 2 to 5 years, have a little older than the severe malaria, have a peak. And then that also goes down as the children grow up. So suggesting that children following repeated exposure are developing immunity to malaria, to clinical malaria. And the pink curve shows you asymptomatic infections. What that means is that, that these individuals have parasites in their blood, but they're not having any symptoms of clinical malaria. And even that, the rate of asymptomatic infections goes down, which means this is really the parasite rate. So there is also evidence of building up anti-parasite immunity, although you never get sterilizing immunity, so that even when you're 30 to 50 years old, there's a chance that you might still get infected, but a lower chance of you getting sick from that infection of malaria parasites. So there's clear evidence for the development of immunity. So if you only understood what are the correlates of this immunity? Is it antibodies? If so, against what antigens? Or are there other arms of the immune system that are coming into play to achieve this? Then you could try and trigger those immune responses using a vaccine. Next, in experiments that were done, some of them as early as in the 60s, it was shown that if you take antibodies from adults living in a malaria endemic area, so people who are sort of in this age group and this kind of, have immunity against both clinical malaria and parasites, if you transfer these antibodies to an infected individual, you can actually clear parasites, suggesting that antibodies do play an important role in this anti-parasite immunity. Now that is blood stage immunity. In terms of pre-erythrocytic stage immunity, again there's data from initially in mosquitoes, in rodent malarias, where immunization with irradiated sporozoites in classic experiments done by the Nussensweigs in New York they showed that if you immunize mice with irradiated Berghia sporozoites and then challenge them with wild-type Berghia sporozoites, the immunized mice are immune. And later, this work was extended to humans. So with irradiated falciparum sporozoites, you can also, again, elicit protective immunity. Suggesting again that there are immune mechanisms against pre-erythrocytic stages that are elicited, and if you understood those, you could think of making subunit vaccines based on antigens from sporozoites. And actually, following this, we already have one subunit vaccine that is well along in clinical development, which is RTS S, which I'll talk to you about briefly. So those are the evidences that suggest that it should be feasible, if you understand your immunology, if you understand the immune responses that lead to protection in malaria, that you could then trigger those immune responses with a vaccine and achieve protection. So what are the different approaches you can take to do that? Well, these are the different stages at which you can intervene. So you could target sporozoites with antibodies, and these antibodies could block the infection of hepatocytes by these sporozoites. Alternately, once the sporozoites are in the hepatocytes, you could try and trigger cellular immune responses, so cytotoxic T cell responses, either by killing with cytotoxic T cells or through the secretion of cytokines. You could clear infected hepatocytes. In the next stage, once the merozoites come out in the bloodstream, you could target merozoites and target antigens that are involved in binding and invasion of red blood cells to try and elicit antibodies that will block red blood cell invasion. And this is the kind of work we do in our lab. Although red blood cells have no MHC class presentation, some of these antigens could also be taken up by other antigen presenting cells, and there could be a role also for cellular immune responses in killing the infected reds, in killing blood stage parasites. And then there is the role of a mechanism referred to as antibody dependent cellular inhibition, where cytophilic immunoglobulins could bind to merozoites, and these could be opsonized by monocytes or even killed again by secretion of cytokines. So these are the different mechanisms. So you could target sporozoites, you could target infected hepatocytes, or you can target the asexual stages, the blood stages. And in terms of trying to block transmission, what you can do is target antigens on the mosquito stages, which means either on the gametocytes, or on the eukinete, and block the development of the parasites in the mosquito. And it's been shown in very elegant experiments that with antigens either on the zygote or eukinete, if you immunize laboratory animals with these antigens and then feed these antibodies against these two mosquitoes through a membrane feeding assay, and along with the parasites, then the antibodies can be taken up and do their job in the mosquito midgut to block transmission. So those are referred to as transmission blocking vaccines. So these are the approaches that people have basically been trying. Now, one of the problems, or two main problems that has been a hindrance to vaccine development is one, we still don't know we don't have the immune correlates. We lack surrogate measures of protection. So we know some people are protected, but we don't have a clear idea of the immune correlates of protection. So we don't know what we are going to try, what we need to achieve to get protection. And the other big problem is the lack of a predictive animal model, which means, we've been able to protect mice from rodent malarias for 20 years, but that has not translated into a viable vaccine in humans. So you may get something that works in mice models, you may get something that works in even non-human primate models, but that doesn't always translate into a vaccine that works in humans. And so because of that, all the vaccine testing has to be done in humans, and that makes it much more expensive and time consuming. So those are some of the approaches and some of the challenges. Now, what has been done so far? Let's take a look at the sort of state of the art and state of the field. Well, as I mentioned, this vaccine candidate that has gone furthest in clinical development, as you're, I'm sure, all aware, is a vaccine being developed by GlaxoSmithKline, originally started off with a collaboration with the Walter Reed Army Institute of Research, and the vaccine is referred to as RTS S. And RTS S is composed of a part of the circumsporozoite protein, not the full length, but a part of the repeats and the C-terminal end of the circumsporozoite protein, which is expressed as a recombinant protein as a fusion with the hepatitis B surface antigen. And that is then mixed with a hepatitis B surface antigen itself, and it forms these virus-like particles, which are then formulated with an adjuvant developed by GlaxoSmithKline called AS01, and the adjuvant is critical to the success of this vaccine, and that's what's used for immunization. A lot of the early development used phase 2A challenge models, which means they immunized human volunteers and then challenged them with infected mosquitoes carrying wild-type parasites, and then followed them over the next few weeks to see if they came down with, if they got malaria parasites in their blood. And a series, and this challenge trial has really been key, development of this challenge model has really been key in allowing them to screen a wide range of adjuvants, and they found out of all the adjuvants they tried, only this adjuvant, actually they had an earlier form called AS02, which is now AS01, but it's basically the same active components, and that it's only with this adjuvant that this vaccine works. And in various trials, they got 30 to 80% efficacy against sporozoite challenge. They then moved to the field, and a number of trials, phase 2B trials, so phase 2A refers to sporozoite challenge in human volunteers. Phase 2B refers to natural challenge in the field. And they found in a series of phase 2B trials that children were protected 30 to 50% against clinical malaria. And they also had early hints suggesting that there was efficacy against severe malaria. Following this, they were encouraged to launch a large phase 3 trial, which was carried out in 5 to 17-month-old children, and also in a younger age group of, I think, 4 weeks to 12 weeks. And at least in the 5 to 17-month-old age group, they had, again, they could reproduce this efficacy. So there is about 50% efficacy against clinical malaria over 1 year. It starts dropping off slowly, but importantly, there was also 45% efficacy against severe malaria. So that's where things stand in terms of RTSS. So we do have a vaccine that at least works partially. Is this good enough to roll out in Africa? That's something that one could debate. But it's actually, out of all the three major problems, the human health problems, HIV, TB, and malaria, in malaria, you at least have a beginning. You have something that works 50%. What can we do to improve on this? Ideally, we'd like to be up at 80 to 90%, but you at least have something that works 50%, and that's actually very good for the field. There have been other efforts to develop pre-erythrocytic stage vaccines using other molecules such as TRAP, multiple epitopes from various pre-erythrocytic stage antigens, liver stage antigen 1. This is a vivax circumsporozoite protein just produced as a recombinant protein, not as a virus-like particle. But none of these have really given any encouraging results. So RTSS has been the only sort of success story in terms of pre-erythrocytic stage vaccines. Except Steve Hoffman sort of picked up on the idea that with irradiated sporozoites, you can actually elicit protection. And that's how most vaccines are made. Attenuate the pathogen or kill the pathogen and use it as a vaccine. And he took on the challenge, it's really, you have to be somebody like Steve Hoffman to take on something like this, which is, he said, I'll build a facility, I'll build a plant to produce irradiated sporozoites. and he actually built a GMP facility to produce irradiated sporozoites. In the first trials they did, this should be phase 2A, not phase 1. Phase 1 is a safety trial. In the first trial where they gave these irradiated sporozoites intramuscularly, they got 0 protection, but he followed that up by using the intravenous delivery route for immunization, and they got complete protection. So irradiated sporozoites, so we actually have in principle a vaccine for falciparum malaria with very, very high protection, which is the use of irradiated falciparum sporozoites. It has been demonstrated that these can be made in a GMP environment that has satisfied FDA. But I see 2 issues with this. One is it has to be given intravenously. And second, can this really be scaled up to produce millions and millions of doses and supplied all across Africa? But in principle, we do have a vaccine, and it'd be interesting to see how this is developed further. So what have we learned in terms of lessons in all this work on pre-erythrocytic stage vaccines? Well, one important lesson is the CSP, circumsporozoid protein, has clearly been validated as a vaccine candidate, which on its own, in a particular context, delivered as a fusion with hepatitis B surface antigen and with a particular adjuvant, can elicit about 50% protection, which is a good beginning. It has demonstrated how you can effectively use the phase 2A challenge model to develop vaccines, to test out formulations, to test out schedules, to evaluate vaccine candidates. What this has also shown is the key role that delivery systems and adjuvants play. The CSP delivered just as a recombinant protein doesn't work. It has to be in a particular context, and that's, so I think developing, in addition to doing the work on malaria, we need to either work with other vaccine developers who may have interesting delivery systems and adjuvants, and that might be key to developing or improving on the vaccine efficacy that has been achieved so far. Few of the problems I see is, one is, even though RTS S works, we still don't have a clear idea of how it works. We don't know what's exactly the mechanism of protection. There are some clues suggesting that it's antibodies against the repeats that are critical, but there's no clear evidences. There's no neutralization titer, for example, that has been identified that needs to be achieved to get protection. So not knowing the mechanism of protection makes it difficult to improve efficacy. You don't know what you need to do to make it better. And the other thing is, if thinking in terms of a vaccine for eradication, what remains to be seen is whether this has any effect on transmission. My guess is probably not, given that the efficacy is only 50%. Next, let's see what we have done with blood stage vaccines or erythrocytic stage vaccines. So remember here, the idea is you try and target merozoite proteins, either surface proteins or apical organelle proteins that are involved in binding to the red cell or are involved in the process of invasion. And with antibodies to these proteins, you try and inhibit red blood cell invasion. The leading candidates for blood stage vaccines were the merozoite surface protein 1, the 42 kilodalton C-terminal end, and the apical merozoite antigen, which is located in the micronemes and is secreted to the surface during the time of invasion. And there were big hopes for these vaccines. A number of groups, the NIH group, the Walter Reed group, have tested them and have taken them into phase 2B trials, tested in children in Africa, and there has been 0 protection. So very disappointing results. And this has been tested with strong adjuvants like ASO2A. So currently, we don't really have any better adjuvants than this. So we can't say, that this will improve if we use a better adjuvant. We don't have one. What could be the reasons for this really poor result, this no efficacy in field trials? When one thing is clearly, malaria antigens are very poorly immunogenic, and you need strong adjuvants. But even with these adjuvants, these antigens actually, especially for MSP1, I won't say that for MA1, but we have never really been able to get very high invasion inhibitory rates with the antibodies that have been elicited in humans. So that's one problem. The second problem is polymorphisms. So AMA1, even if you have very high invasion inhibitory antibodies, works only against the homologous strain. If you have a heterologous strain with polymorphisms in AMA1, then the antibodies don't do as well in terms of inhibiting invasion. So the other big problem is sequence polymorphisms in target antigens. And I think that's the lesson we have learned from these two studies, which is one, you need to really get very high growth inhibitory antibodies before you take something to the field, and second, you must address the problem of sequence polymorphism. There are other efforts, other antigens that are currently in the pipeline. This is a fusion protein of MSP3 and MSP119, which is being developed actually by a colleague at ICGEB, Dr. Chauhan's group. Our group, as well as a number of others, are working on some other antigens called the RH proteins from the ruptrees, which are actually very well conserved. The apical asparagine-rich protein, another ruptree protein being developed at ICGEB, and this is the Duffy binding protein, which my group is working on from Vivax. So there are other antigens that are under development for blood stages. But I think what we must learn from our failures is that you need to achieve high invasion inhibitory antibodies, address the problem of polymorphisms. Single antigens are unlikely to work. So we need to find combinations of these antigens that give you synergy and high growth inhibition. And again, we don't have correlates of immune protection. We don't know against which antigen you need antibodies to get that protection and what those antibody levels need to be. And so there's more work to be done in terms of field studies. Let's go next to the transmission blocking vaccine candidates that have been worked on. The two leading candidates are PFS25 and PVS25. These are both eukinete proteins, and it's been shown these have been developed at the NIH, that antibodies to these proteins will be picked up by the mosquitoes and they will block transmission, block the development of oocysts in the mosquito. Again, you need to get very high growth, high transmission blocking activity with those antibodies, and to achieve that, the NIH group used a very strong adjuvant called Montanide ISA51, which is basically incomplete Freund's adjuvant. But when you use such a strong adjuvant, you have problems of reactogenicity. And in fact, the trial that they were doing had to be stopped due to reactogenicity problems. And if you use a more human compatible adjuvant such as alhydrogel, then you don't get the transmission blocking activity you need to get the high rates of inhibition. Other candidates that are being developed, PFS 4845, and this interesting candidate from Johns Hopkins, which actually targets a mosquito's midgut antigen. This is probably an antigen that the parasite needs to bind to cross the midgut, and if you target it with antibodies, then you probably block that interaction and you can block transmission. So one can think of not only antigens on the gametocyte or on the eukinete, but you could also target mosquito midgut antigens to block the transmission stages from developing. So what we have learned here is you really, just like the invasion inhibitory antibodies for the asexual stages, for transmission blocking also, you need to achieve high transmission blocking activity, and you have to do this with human compatible adjuvants, and there aren't many that are both safe to use and give you that high antibody titers. And so instead of recombinant proteins, you need to look at other delivery systems. One such system could be viral delivery, with the prime boost, which is what Adrian Hill is doing. Alternatively, one could look at VLPs, virus-like particles. So there's a lot of work to be done in now combining what we have sort of discovered in the malaria field with discoveries from related fields in vaccine development to try and come up with formulations that will be immunogenic. And you need to, here fortunately we have a very nice assay, a transmission blocking assay, which can be done quite routinely, which can guide vaccine development. But we need to find ways to integrate this with clinical development strategies to develop these vaccines quickly. So that's what we've been doing so far. If we are to develop, now we are saying we want to use vaccines and see how they can help in the eradication effort. So what changes in terms of our thinking? What needs to change in terms of our thinking? Well, one thing that has clearly changed is the context. There was a time when we were developing these vaccines for morbidity and reduced mortality when malaria was 500 million cases and 1 million deaths. Now, with the intensive implementation of malaria control tools, we have dramatically changed malaria incidence rates in many regions. There are countries, as we heard earlier, Mexico, where we are, and Ecuador, many countries in this part of the world that are actually heading towards elimination. So will a vaccine that targets reducing morbidity and mortality be of interest to these countries? Probably not, unless it also helps them to reduce transmission. So instead of vaccines that only reduce morbidity and mortality, we need vaccines that will also have an effect on transmission. And so far, as you see, we've been thinking of these two things separately, either vaccines that reduce morbidity and mortality or vaccines that reduce transmission, which we call transmission blocking vaccines. And through this consultative process of Malaria, which produced that, all the articles in that book or that journal that was handed out, we came up with the idea of thinking of vaccines that interrupt malaria transmission. And I'll explain to you what these are, what these could be. So clearly, our focus needs to start bringing in the idea of reducing malaria transmission as a central part of our vaccine development effort. And second, importantly, we can't be thinking just of falciparum alone. We need to think of vivax. Parts of the world heading towards elimination, again, like this part of the world, are not, they need a vaccine also for vivax. So this is what the vaccines that interrupt malaria transmission, a new way of thinking of vaccines that need to be developed in response to the eradication agenda. These are the things that are important. First, the target is really to reduce transmission. And that is often expressed in a simple way by saying that we need to achieve an R0 less than 1. What is R0? R0 is the basic reproductive rate. That is, if you have one infected person, how many people end up with an infection because of that one person? If the number is more than 1, then the infection spreads. If the number is less than 1, then the infection or that infectious disease sort of basically gets burnt out. And so anywhere where you have ongoing transmission of malaria, R0, that's because R0 is more than 1. If you drive it to R0 less than 1, then over a period of time, that infectious agent will become extinct. So classically, it's been thought of as classical transmission blocking vaccines, as well as the pre-erythrocytic and asexual stage vaccines, that is VIMTS, or these vaccines that interrupt malaria transmission, could encompass both. Certainly they could encompass transmission blocking vaccines because their primary purpose is to reduce transmission, but they could also encompass pre-erythrocytic vaccines and asexual stage vaccines as long as they have a dramatic effect on reducing parasite densities, and not just parasite densities of the asexual stages, but also if this leads to reducing gametocyte densities in the population. If not, then they would not qualify as vaccines that would be useful for reducing malaria transmission. One could also think of in VIMTS that we have a transmission blocking component, and to that you add either a pre-erythrocytic and asexual stage vaccine, because that would give the added benefit of protecting individuals from clinical malaria in case malaria is reintroduced, in case there is a reversal. And now you'll have a population, even adults, who have no immunity because malaria transmission rates were already very low. So that's the kind of vaccines you want to develop. So the primary goal is have vaccines that will have an effect on reducing transmission, but you could have an added component that also takes case of clinical malaria. So one way to sort of focus your thinking on terms of what will our vaccine achieve, what will it look like, is in industry talked of as developing a target product profile. And we'll go through one just to give you a sense of how one goes about developing these target product profiles. And once you have a target product profile, then the question you need to ask is, how will you go about developing a vaccine with this target product profile? What is the R&D you require from discovery to the following stages? And how will you evaluate this vaccine? So what will be your clinical development and registration strategy for such a vaccine? So a target product profile, when you try and develop, you always target, you have what is a desired target. This is the ideal. This is if somebody said you have a wish, and what would you desire if you could get anything you wanted? That's the desired target, but you have to come back down to reality and also agree on something you will minimally accept. So this is the minimally acceptable target. That is, if you get this, you'll be okay. So let's go through this. So indication, what will this vaccine be used for? The desired target is interruption of transmission of all Plasmodium species. Ideally, I'd like to get rid of all malaria. We're talking of malaria eradication, not just falciparum. But to begin with, you're willing to accept a vaccine that interrupts transmission of P. falciparum, but all strains of P. falciparum. What's the target population? All age groups, including pregnant women and immunodeficient persons. That's your ideal, but you'll be willing to accept if you can just target healthy persons, including infants, children, and adults. It's important that this vaccine will be used widely, so you definitely need to include infants, children, and adults. You could leave out pregnant women to begin with, given there may be safety issues. That's anyway a very small, it's a small part of the total population, and you can leave out immunodeficient persons. Route of administration, how will you deliver this vaccine? Ideally orally or IM or subcutaneous. Orally would be challenging, so you can leave that out in your minimally acceptable target. Product presentation, ideally it would be simplest to deliver if it was a single dose, auto-disposable pre-filled device, but that's again, single dose is asking a lot from a vaccine. You would be willing to accept a liquid form. Also, having a vaccine where it's pre-formulated can be quite challenging. So even if you have a vaccine where the antigen and adjuvant are in separate containers, you'd be willing to go with that. The dosage schedule, single dose, no booster required, but realistically, even if you have 2 doses and a booster required 4 to 6 months after the second dose, so 3 doses, that should be fine too. Next, expected efficacy reduces R0 to less than 1 for at least 2 years, but you're willing to accept that. And here, desired, that is, even if you have in a place no other control efforts, just your vaccine, that would be great. If you just with the vaccine, you can reduce R0 to less than 1. But realistically, you're willing to accept that if you have a vaccine where you reduce R0 to less than 1 for 1 year in conjunction with other measures. Co-administration, you clearly don't want to affect responses, immune responses to other EPI vaccines that children normally get, but you're willing to accept, that is, you could give it along with the EPI vaccines, but you'd be willing to accept that this will have to be given separately, but you still don't want to have any effect on the EPI vaccines. Shelf life, storage temperature, registration, etc. So this is how you go about. So once you have this target product profile, you then, whatever you develop must meet this. So it's something that guides your development effort. What do you need to do to achieve this? Well, first of all, clearly, you need to expand your research portfolio to include Plasmodium vivax. Second, how are these vaccines going to work? They're going to work by interrupting transmission, and you can do this in 2 ways. One is by interfering in the development of the parasite in the mosquito, or by reducing the gametocide load within the population. So even a very successful, very highly efficacious pre-erythrocytic stage vaccine could reduce the gametocyte load and could have a role in reducing transmission. With respect to blood stage vaccines, it's still an open question. A highly effective blood stage vaccine may or may not actually affect your gametocyte load. And if it doesn't, or you can even think of a scenario where the parasite actually as a response makes more gametocytes, then you might actually make transmission worse. So it's something that you must keep an eye on if you're developing blood stage vaccines. To achieve this, what you need is you certainly need we have a pipeline, as I showed you, of candidates. For actually you have a reasonably rich pipeline for blood stage vaccines, not so many in the pre-erythrocytic stages, and you do have some in the transmission blocking stages, but you certainly need more work to understand host parasite and vector parasite interactions to come up with new candidates. And these new candidates need to be tested not just individually, but in combinations to see if you get synergy and very high inhibition of invasion or oocyst development or transmission blocking, because that's what you really need to make an effective vaccine. This I already discussed. For blood stage vaccines, keep an eye on whether you really reduce gametocyte densities and infectivity rates. And next, we need to understand how reducing gametocyte rates or how these vaccines actually affect individual infectivity and how that affects transmission rates at a community level. And so there's very little data on this so far, and that's something we need to understand, because this will actually help you design trials for studying reduction in transmission. And that's not going to be very easy. As Lee mentioned, a way to sort of do things efficiently is to have the data, to understand the biology, have the data, so that you design trials in the right sort of way to get your answers to move along the development process. Again, identification of surrogates at individual level. You see, it's very difficult to measure transmission rates at a community level. But if you can make the link in a few studies to see how individual infectivity rates are related to transmission rates at a community level, then one could measure more easily the individual infectivity rates and predict, using mathematical modeling, what happens at the community level. And one can also think of vaccines that target the vector, as we discussed earlier. Next, once you have your antigens, you have your assays done, and you have things that look worth taking into the clinic, then you need to have a clinical development and regulatory strategy. And that's really the more downstream, translational end of things. And again, any clinical development strategy, if you're going to develop a vaccine that's going to be used for reducing transmission, that has to be the critical test. Is your vaccine reducing transmission? These kinds of vaccines will really be used widely, and so they must be very well tolerated. They have to be able to be used in infants, in children, in adults, and we have to find ways to make them really immunogenic, give you very high antibody titers that are long-lasting. And as I mentioned, identify surrogate measures so that you can measure infectivity or effects on infectivity at the individual level and to predict what the efficacy will be at the population level. And a lot of this might require close interactions with regulatory bodies. They must approve what you are trying to do. So, and a lot of this work, of course, will be done in endemic countries. So you need to engage with endemic country scientists, with endemic country regulatory agencies, and this is where WHO could play a major role. So that's where I'll end. So what I've tried to explain to you is give you a sense of where we are in terms of vaccine development today, and that was based on really developing vaccines that target morbidity and mortality, and how we need to move to now vaccines that will have an impact on transmission if we want these vaccines to be used in the elimination, eradication agenda. So this work was actually, this took a lot of interactions, and this was the sort of core group of people who met at least 3 times to discuss this and came up with the paper that is in that book. on developing vaccines to interrupt malaria transmission. We also did a number of consultations more widely with the malaria research community, and I had at least 3 meetings with them to solicit ideas, suggestions, and Matiana had helped us draft all this, and this was all funded by the Gates Fund.